Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science.
And if you appreciate these deep dives, which are, let's be honest, your shortcut to sounding brilliant, please just take a second right now.
Yeah, hit like, subscribe, and share this with your friends and family. It really does support the Sand Science Experience and lets us keep bringing you this stuff.
Absolutely.
So today, we are embarking on a deep dive that, and I'm not exaggerating, completely rewrites the cellular rulebook for how mitochondria can trigger inflammation.
It really does. We're looking at a study that found this totally unexpected link between how mitochondria are regulated, how cells die, and even neurodegenerative diseases.
Specifically Parkinson's, which is just a huge connection to make.
A huge connection. Our main subject is a protein called MAPL. That's MAPL, mitochondrial anchored protein ligase.
Right. And MAPL was always known as a sort of quiet manager of cell death.
A very quiet one. But the new research shows it has this secret life. It's the conductor of a really explosive, very inflammatory death pathway.
So our mission today is to map out this brand new mitochondrial lysosome axis.
And it's not just a new pathway. It's a whole new type of death that MAPL is in charge of.
We're moving from apoptosis, which is that quiet, programmed cell suicide,
To something much louder, something called pyroptosis.
which literally means fiery death.
Right, exactly. Apoptosis is neat, it's clean. Pyroptosis is messy, it's explosive. The cell literally ruptures and spills inflammatory signals everywhere.
So we're going to trace the molecular handoffs that let a protein on the mitochondrial surface become a trigger for a kind of self-destruction that has direct links to Parkinson's genes.
It's an amazing piece of molecular choreography.
Okay, let's start with the star player, MAPL. It's a sumo E3 ligase, sitting right there on the outer mitochondrial membrane. For anyone who's a bit rusty on their cell bio, what exactly does a ligase like this do?
Think of it like a tagging gun. MAPL's job is to attach these little protein tags, called sumo tags, onto other proteins.
And that tag changes what the other protein does.
Completely. The old thinking was that MAPL's main job was to tag a protein called DRP1.
DRP1 helps mitochondria divide.
Right. By tagging it, MAPL would sort of stabilize the mitochondria, making it easier for two other proteins, BX and BEC, to form pores and release cytochrome C.
And that would lead to that classic quiet apoptosis.
That was the textbook model.
But this new study just blew that whole model up, didn't it?
It did. They did the definitive experiment. They used cell lines that genetically lacked BX and BEC.
So the two proteins that are absolutely required for that old pathway.
Mandatory for it. So if MAPL needed them, nothing should happen in these cells.
Yeah.
And yet, when they overexpressed MAPL, the cells still died. They even showed signs of active casposes.
Wow. So it's undeniable proof.
It's absolute proof. MAPL was using a completely different road to get to the same destination of cell death.
And that new road is pyroptosis. So why is this fiery death so much more dangerous to all the surrounding tissue?
Because it's an alarm bell. It's designed to be. Apoptosis is like a quiet demolition. It neatly packages everything up for disposal.
No fuss, no mess.
Pyroptosis is like setting off dynamite. It activates this huge inflammatory sensor complex called the NLRP3 inflammasome.
So the cell doesn't just die, it screams on its way out.
It screams and it throws all its inflammatory cargo cytokines, all its contents, into the environment as it bursts. It incites inflammation in all its neighbors.
And the researchers found that MAPL's activity, specifically its ability to tag other proteins, was what started this whole fire.
100%. The switch depends on its ring domain. That's the functional tagging gun part of MAPL.
Okay.
And when that's active, MAPL starts driving this early inflammatory response, turning up genes like IL-6, IL-1B, and even the NLRP3 sensor itself.
It's basically flipping the main circuit breaker for the cell's internal immune system.
That's a perfect way to put it.
Okay, this is where it gets really cool. For a cell to go into full pyroptosis mode, it needs a danger signal. Something has to be released into the main part of the cell, the cytosol.
And that critical ingredient, the thing MAPL is pushing out, is mitochondrial DNA, mtDNA.
mtDNA is the fuel for the fire.
It's the absolute key. The cell's innate immune system is trained to ignore our own nuclear DNA, but if it sees DNA floating free from a mitochondrion...
It thinks it's an invader, a virus or a bacteria.
Exactly. It panics.
So how did they prove that mtDNA was the linchpin here?
Oh, this was brilliant control. They used cells that had been completely stripped of their mitochondrial DNA. We call them row cells.
No mtDNA at all.
Zero. And when they activated MAPL in these row cells, nothing happened.
They were completely protected.
Completely. No mtDNA, no inflammatory death. That proved MAPL's kill switch was entirely dependent on getting that specific mitochondrial cargo out.
Which brings us to the next massive puzzle. How does a protein on the surface of the mitochondria push DNA out from the very inside? There are two membranes in the way.
Right, it's not just a passive leak. This is an active transport system. It uses these tiny little packages called mitochondrial-derived vesicles, or MDVs.
Little shipping containers.
Perfect analogy. MAPL basically commissions these MDVs to be loaded up with mtDNA and launched out into the cytosol.
So MAPL is running an express shipping service for its own toxic cargo. How does it physically pinch off these vesicles?
It uses a couple of proteins called the MARO GT passes, AMARO1 and MARO2.
Aren't they usually involved in just moving the whole mitochondria around?
Yeah, they're like the logistics crew. But here, they help bend and pinch the mitochondrial surface to form and cut off these MDVs.
And what's so fascinating is how selective these MDVs are. They're not just random bits of mitochondrial debris.
Not at all. These are highly specialized packages. When they looked inside, they found mtDNA, and they also found bits of the inner mitochondrial membrane, like complex 1.
Okay.
But, and here's the key, they completely lacked a marker from the outer mitochondrial membrane, a protein called TOM40.
Which tells you it's a very specific cargo-selected process.
Yeah.
A deliberate signal, not just random damage.
Exactly. It's targeted shipping.
Now that the package is sent, where is it going? Where are these inflammatory MDVs being delivered?
They're being sent to the endolysosomal compartment.
The Cells Recycling and Waste Disposal Center. The lysosome.
That's the one.
Yeah.
The MDVs dock there and deliver their mtDNA right into the lysosome. They saw about 12% of lysosomes ended up with DNA inside them after MAPL was turned on.
And they were careful to check it wasn't just normal mitophagy, right?
Oh, yeah. They ruled that out completely. This isn't the cell getting rid of a whole damaged mitochondrion. This is a tiny targeted delivery of a specific signal.
So now we have our inflammatory trigger, the mtDNA, locked inside the lysosome, which is basically a vault designed to keep dangerous stuff contained. This brings us to the central mystery of the paper. How does that mtDNA get out of the lysosome to sound the final alarm?
The vault has to be breached. They watched for this breach using a marker protein called galectin-3, or GAL3.
And GAL3 basically rushes to the scene of any crime, any damaged internal membrane.
Exactly. It's like a first responder. And sure enough, when MAPL was active, they saw GAL3 swarm all over the lysosomal membranes.
A physical rupture.
A physical rupture, and it was dependent on MAPL's tagging activity.
And the tool used for the breach.
Yeah.
This is where the executioners of Pyroptosis, the Gasdermins, show up in a place nobody expected.
This is a huge paradigm shift, a really big deal for cell biology. Gasdermin proteins are famous for one thing, punching holes in the cell's outer wall, the plasma membrane.
That's how they kill the cell, the final explosion.
That's what we thought. But this study found that GSDME, Gasdermin E, was actually the one responsible for rupturing the lysosomal membrane first.
Wait, so this protein, famous for blowing up the whole cell, is actually starting the sabotage from the inside by puncturing the recycling bin?
Yes, exactly. It's an inside job. When they silenced GSDME, the breach was prevented. The GAL3 alarm signal it never went off.
Yeah.
And they proved it biochemically, too. They actually isolated the active, pore-forming part of GSDME and found it physically stuck in the lysosomal membranes.
Credible.
And this also cleared up the chain of command. Losing GSDME also blocked the next step, the cleavage of GSDMD, which is the gasdermin that blows up the whole cell.
So GSDME and the lysosomal breach are definitely upstream. It's the first domino.
It's the first domino.
So once that lysosomal wall is breached, the mtDNA is finally free. It floods into the cytosol, and that is the starting gun.
That's the starting gun for the whole inflammatory cascade. The mtDNA is immediately detected by a sensor called cGAS.
The cGA sting axis.
The cGA sting axis. cGAS sounds the alarm, sting amplifies it, and that's what drives the full pyroptotic explosion that finally kills the cell. If you block cGAS, you stop the death.
This whole pathway is it's so intricate. But then the study connects it all directly to Parkinson's disease. This gives it that huge clinical relevance.
It really does. It connects these deep cellular mechanics to actual genetic risk factors. The initial screen they ran to find things that could stop this MAPL-induced death.
Yeah.
It kept flagging two very famous Parkinson's genes, VPS35 and LRRK2.
And what's so cool is that they act at totally different points in this whole process.
Completely different stages.
Okay, let's take VPS35 first. Where does it fit in?
VPS35 is an early player. It's all about logistics. When they got rid of VPS35, the cells were saved from death, and they saw far fewer of those mtDNA packages forming.
So VPS35 is required for the initial step, the packaging and delivery of the MDVs to the lysosome.
Right. No VPS35, the toxic package never gets sent, and the whole pathway just starts before it can even really begin.
And LRRK2, the other more notorious Parkinson's gene, it acts later, at the vault breach stage.
Exactly. When they silenced LRRK2, it had no effect on the delivery. The mtDNA packages still arrived at the lysosome just fine.
Okay, so the delivery is fine.
But the breach itself was significantly reduced. That GAL3 alarm signal was much, much weaker.
So LRRK2 is somehow required for the GSDME-driven rupture of the lysosome.
It seems to be. So you have this terrifyingly elegant sequence.
Let me see if I've got this. MAPL starts the process. VPS35 is needed to package and ship the mtDNA. The package gets to the lysosome, and then LRRK2 is needed to help GSDME punch the hole to let the trigger out.
You've got it. That seems to be the sequence. LRRK2 facilitates the physical leakage that unleashes the mtDNA.
And did they confirm this holds up in actual immune cells? Since this is all about inflammation?
They did. They used bone marrow-derived macrophages, BMDMs, and the pathway is totally conserved.
So getting rid of MAPL protected them from pyroptosis.
Protected them beautifully from standard immune triggers like LPS and IF and gamma. It confirms this is a real, relevant inflammatory pathway.
And I'm guessing the final nail in the coffin was that cGAS sting experiment.
Oh, that sealed the deal. They took their protected cells, the ones without MAPL, and they just skipped all the early steps. They added the product of cGAS directly into the cells.
They bypassed the mtDNA release.
Exactly. And that completely reversed the protection. The cells were instantly resensitized to death. It's the definitive proof that MAPL's main job here is just getting that mtDNA out into the open.
This has been a really stunning deep dive. So to recap for everyone listening,
Go for it.
MAPL, this protein on the mitochondria, orchestrates a new mitochondrial lysosome pathway. It actively packages mtDNA into vesicles, a process needing the Parkinson's gene VPS35.
These packages are delivered to the lysosome.
Whereas a process dependent on another Parkinson's gene, LRRK2, the GSDME protein, punches a hole in the lysosome.
Which releases the mtDNA into the cytosol, triggering that lethal pyroptotic cascade through cGS sting.
It's just a masterpiece of a mechanism.
And the therapeutic potential here is what's so fascinating. The discovery that GSDME punching that hole seems to be dependent on LRRK2. That's the big actionable takeaway.
Right. Before this, we knew LRRK2 was bad, but we didn't know it was tied so directly to the physical act of inflammatory cell death.
It opens up a totally new angle.
So it suggests that targeting LRRK2 might not just be about general neuronal health, but could specifically stop this inflammatory self-destruction that might be at the heart of diseases like Parkinson's.
It's possible. I think future research is going to focus on exactly that. How does LRRK2 modulate that physical rupture of the lysosome to control inflammatory cell death?
It just shows how deeply interconnected mitochondrial health, innate immunity, and neurodegeneration really are.
It's all one conversation at the molecular level.
Truly astounding work. Thank you so much for breaking that down with us.
My pleasure.
And for everyone listening, remember to like, subscribe, and share this with your friends and family. Help us continue the Sand Science experience.